This morning on the Arabidopsis Research Roundup, I'm joined by Professor David Salt from Aberdeen University, and we're going to be talking about a recent paper they've had in PLoS-Genetics in collaboration with some Swedish collaborators. And the title of the paper is quite long, but I'll try and get through it. The multi-allelic genetic architecture of a Variance-Heterogeneity locus for molybdenum concentration in leaves acts as a source of unexplained additive genetic variance. So, good morning, David. I hope I got through the title okay there.
Yeah, it's horrendously long. It's almost an abstract in itself.
It is, rather, it is. So can you give us an explanation about what the paper's all about?
Yeah, I'll try to simplify this a little bit. So this is work that actually I've done in collaboration with Örjan Karlsberg at the Swedish University of Agriculture in Uppsala. So Örjan takes the lead on this paper. He's the kind of population geneticist behind it. But I think I'll try to boil it down into a nutshell. So the key piece about the paper is to try to do a new kind of genome-wide association mapping. Mm-hmm. And so I think maybe for me to explain why it's new, I probably just need to briefly explain what a regular genome-wide association mapping tries to achieve. And so basically, in regular genome-wide association mapping, what you're doing is you're taking a genome in which you've painted on multiple thousands, if not millions, of known markers. And these markers are biallelic, so they can come in one form or another form. And they're a SNP marker, so an A or a T, for example. And they're equally dispersed across the genome. And different populations of Arabidopsis thaliana are being developed, which have between 250,000 up to 10 million SNPs that have been tagged across the genome of up to 1,200 different accessions. And so in this particular study, we used a set of 360 accessions. And the way that genome-wide association mapping basically works is if you can imagine that you simply step across the genome and you look at each individual marker, and what you do is you take the 360 plants that you phenotyped. And for us, our focus is on the ionome, and particularly in this paper, focusing on the accumulation of molybdenum. Yep. Essential micronutrient important for a couple of critical processes in plants, including nitrogen assimilation, ABA biosynthesis, etc. And so you take this set of 360 plants, and what we did is we measured the molybdenum levels in the leaves of plants all grown in kind of a common garden in the greenhouse. Mm-hmm. And then what you can do for the genome-wide association mapping, if you imagine what you do, you go to the first SNP, and you essentially take your 360 accessions and you divide them up into the individuals that have one version of the SNP and into individuals that have the other version of the SNP. So you end up with basically two piles of plants, if you like, for each of those, for that first SNP. And then what you do is you simply take the mean of one pile and you compare it to the mean of the other pile for the phenotype you're interested in. So for us, that would be molybdenum. Yeah. And you essentially do a simple t-test, and you ask the question, for that particular marker, do you see a significant difference in the molybdenum concentrations between the two forms of that marker? Mm-hmm. Okay? Yep. And that basically gives you a p-value. And what people like to do is they generally kind of plot that as a negative log p, just so that better is bigger, as opposed to if it would just be a standard p-value, then smaller would be bigger. So that gives you those kind of classic Manhattan plots where you see that a big peak going up is good. Yep. And then you basically just do that across the whole genome at each of the markers. And what you're looking for are markers in which the mean is significantly different between the two markers. And that's basically standard kind of genome-wide association mapping. Mm-hmm. There's obviously multiple complexities about population structure, confounding this and things, but I think don't need to worry too much about that. But the basic idea is you look at each of these markers, you make two piles, you look at the mean of the phenotypes, and you ask the question, is there a difference between the means? So it's based on the means. Yeah. So what that kind of process assumes is that each of these SNPs, if you do identify an association, let's say a particular SNP, you find that the accessions, let's say the 60 accessions with one version of the SNP, and they have high molybdenum compared to the rest of the accessions with the other version, which have low molybdenum. What that kind of assumes is that SNP that's associated with high molybdenum is linked to a single allele of some gene which is causing molybdenum to be high. Yeah. But what you could have is a situation where actually that SNP is linked to a region of the genome which actually contains multiple alleles. And some of those alleles may make molybdenum high, and some of them may make them low. But when you just take the mean, all that is kind of lost. That information is lost. And so what this variance GWAS does is it tries to kind of probe that idea to try to identify if there is what's called this allelic heterogeneity. So it's not simply that a SNP is marking an allele which affects molybdenum. It could be that a SNP is marking actually multiple alleles which affect molybdenum in different ways.
Okay.
And so just doing the comparisons on the means doesn't help you there, because you could imagine a situation where one version of the SNP actually tags two alleles. One of those alleles makes molybdenum high, one makes molybdenum low, and the mean comes out looking just like the other version of the SNP. And so you would conclude there's no association, but in fact there is something going on, but you're just not detecting it with the mean. So what we've been working on, what Orjan's been working on with us, is this idea of, instead of using the mean for your genome-wide association mapping, you use the variance.
All right.
So you basically do the same thing. You step along the genome, and at each of the SNPs you make two piles. But instead of comparing the mean of the phenotypes, now you compare the variance. Mm-hmm. And when you do that, you now have the possibility to detect that situation where I said you had a marker which was actually connected to two alleles. One made the molybdenum high, and one made the molybdenum low. If you took the mean, you wouldn't detect that. But if you took the variance, you would. Yeah, because in that situation, that would have a very big variance, because one allele would be making Mo high, the other allele would be making Mo low. The means may look the same, but the variance for one allele would be very high. The variance for the other version of the marker would be lower. So that's basically what this paper does. Yeah. It uses this variance GWAS, and what that allows you to do is to allows you to detect this idea of allelic heterogeneity in genome-wide association mapping. And we used that to actually reanalyze data that we had for molybdenum.
Okay, so from what your description there, the strength of this paper is really using this new technique, and this is going to be applicable for many other people, anyone who's interested in this sort of analysis, to use it for their particular condition of interest, I'm guessing.
Exactly. Yeah, I mean, you can use this to reanalyze existing data or analyze newly collected data. And the idea is it lets you really dissect the genetic basis of traits in more detail, and maybe identify stuff that you may have originally missed just doing GWAS based on the means. And
so you said you did a reanalysis, and you found a couple of new novel genetic loci that are interested. Yeah, okay.
Yeah, super interesting. So in fact, we already knew that there was a strong association of this transporter that we earlier identified called MOT1. Mm-hmm. And we know that this transporter is involved in controlling molybdenum accumulation, though we actually don't really understand functionally how that works. But what we identified with the VGWAS is, in fact, there's actually a linkage block which contains MOT1, and within that linkage block, there's actually two different alleles which are causing variation in the function of MOT1. In fact, one of them is a deletion in the promoter, which causes a reduced function, and the other is an insertion in the promoter, which causes an increased function.
Okay.
And then we also identified two other SNPs that were also affecting molybdenum, also within this linkage block, but in different genes.
Okay, I see.
So we're thinking what might have happened is there may have been an inversion, and then this inversion has then now stopped recombination and allowed this particular haplotype to kind of evolve away from the other version.
Okay, I see. So just finally, so obviously this is a tool that many people are going to be interested in, and from obviously GARNet, we're interested in this community resource. But it also strikes me as being somewhat relatively straightforward to think about it. So as the blockage on developing this has been the computational, has it been a computational blockage, or just having the amount of data available to enable to get enough information to get this variance?
Yeah, I think that's a great question. I mean, I think people have been thinking about this kind of variance heterogeneity long before Orjan started working on this VGWAS. But I think that the limiting step in really getting this to work has probably been the tools, in particular the SNP densities, needed for really doing GWAS. And I think that you could say that about GWAS as a whole. I think that it's only in the last couple of years that we've really moved from just having thousands of SNPs now to millions. On these panels, which now are going to really throw open people's ability to do genome-wide association mapping and actually get really interesting, exciting results.
Absolutely. So we'll encourage people to take a look at the paper and use this technique to look at their conditions of interest. And of course, it highlights nicely a little. I mean, in a year it's going to be, but of course we're going to have have a GARNet organised conference by yourself and Ian in Cambridge on techniques such as this and the power of them for gene discovery and the like as well.
Exactly, and Orjan's going to be speaking there.
Absolutely. Should be great indeed.
Yep,
Lovely.
great.
All right, thank you very much, David, for that.
Brilliant.
Thank you.
No problem.
All right, bye-bye.